Hello friends, welcome in EPG Pathshala. I am Dr. Ashok Sharma, Assistant Professor and Ramalinga Swami Fellow, Department of Biochemistry, All India Institute of Medical Sciences, New Delhi. In previous module, I have introduced you about epigenetics and effects of environmental factors on epigenetic control and its influence on human diseases. In today's module M-32, we will discuss about DNA methylation. In detail, which one of the best extensively studied epigenetic mechanism and play very crucial role in gene regulation. As we discussed in previous module that normal growth, development and differentiation of all the cells are regulated by the genetic and various epigenetic factors acting at the genome. Therefore, with a single genome, we have hundreds of epigenomes. DNA methylation and histone modification are the major epigenetic marks which have been studied widely due to their direct role in regulating the gene expression in normal cell as well as during various human diseases. Furthermore, during the past decade, extensive research has been done on function of DNA methylation and its potential to be used as a therapeutic target in various human diseases including cancer. In this current module, we will understand the following objectives. First, what is DNA methylation? Second, role of DNA methylation in gene regulation. And finally, how to analyze DNA methylation along with potential use of DNA methylation in clinics. So, what is DNA methylation? DNA methylation is a heritable epigenetic mark involving the covalent transfer of a methyl group to the fifth carbon position of the cytosine ring of CpG dinucleotides of DNA from S-adenyl methionine SAM by the enzyme DNA methyl transferase called DNMTs as shown in this figure. During zygote formation, DNA methylation mark is typically removed and then re-established in the embryo at approximately the time of implantation. Most of the DNA methylation mark is essential for normal development and it plays a very crucial role in number of processes. Transcriptional silencing, maintaining the genomic stability by protecting the genome from transposition, and suppression of repetitive elements, line elements, aluminum elements transcription and the transposition, and when dysregulated contributes to disease like cancer, genomic imprinting, X chromosome inactivation, and finally also play very crucial role in tissue specific gene expression. DNA methylation is regulated by a family of DNA methyl transferases called DNMTs enzymes. So, DNMT enzymes are DNMT1, DNMT2, DNMT3A, DNMT3B and DNMT3L. Structure of DNMTs are shown in figure. DNMT1 preferentially methylates hemimethylated DNA in vitro and is localized to replication foci during S phase. DNMT1 is called maintenance methyl transferase because primarily it is responsible for copying DNA methylation pattern to the daughter strand during DNA replication. Mouse model with both alleles of DNMT1 deleted are embryonically lethal at approximately day E9. DNMT2 is a methyltransferase homolog that methylates cytosine 38 in the anticodon loop of aspartic acid transfer RNA instead of DNA. DNMT3A and 3B, in contrast to DNMT1, have preference for unmethylated CpG dinucleotide and perform de novo methylation during development. Overall, mammalian genomes are depleted of CpG sites that may result from the mutagenic potential of methyl cytosine that can deaminate to thymine. On the basis of location and DNA methylation pattern of CpG site, we classified as below: CpG island, CpG source, CpG self, and CpG body methylation. CpG islands are basically most of the genes have an upstream, usually around 1 kb base pair upstream from the start site is GC rich region with higher CpG density called CpG island. CpG source, basically this is a region called CpG source located as far as 2 kb base from the CpG island. And CpG self is the region called CpG self located as far as 4 kb from the CpG islands. And gene body methylation, basically the gene body is considered the region of the gene past the first exon. DNA methylation of the gene body is associated with a higher level of gene expression in dividing cells. This figure has shown the location and pattern of the CpG sites in representative gene in normal and cancer cell. The hypermethylated CpG site is represented by black lollipop, while hypomethylated CpG site represented by white lollipop. Most of the genes have an upstream, usually around 1 kb base pair of the upstream is GC rich region with higher CpG density, as we tell you previous slide, called CpG island. The majority of the gene promoters, roughly 70%, reside within the CpG island. CpG islands are highly conserved between the mice and human. The methylation of CpG islands results in stable silencing of gene expression. During gametogenesis and early embryonic development, CpG islands undergo differential methylation associated gene regulation. The ability of methylation to regulate gene expression through CpG island is particularly important for establishing genomic imprinting, where only one of the two available alleles is expressed in the progeny, while the other allele is repressed by DNA methylation. CpG islands are classified according to the Gardner-Gardner and Fromer definition and Takei and Jones definition. According to the Gardner-Gardner and Fromer definition, the CpG island should be at least 200 base pair long and GC content more than 50%. Observed and CpG and expected CpG ratio is greater than equal to 0.6, while as per Takei and Jones definition, region should be more than 500 base pair, GC content more than 55%, observed CpG and expected CpG ratio greater than equal to 0.65. With these definitions, CpG islands are more likely to be associated with the region of genes and exclude most ALU families. There are about 29,000 such regions in the human genome. DNA methylation may affect the transcription of the gene in two ways. First, the methylation of DNA itself may physically impede the binding of transcriptional protein to the gene, and second, likely more important, methylated DNA may be bound by protein known as methyl CpG binding domain protein or MBD proteins. DNA methylation play very crucial role in to maintain the tissue specific pattern as well as in embryogenesis and tissue differentiation. A set of nucleic acid methylation modification in an organism genome or in particular cell called methylome. In Human Epigenome Project, complete DNA methylome of human are now available, helping clarify the evolutionary story of these epigenetic marks and its distribution in key genomic elements. In non-CpG methylation, a methyl group is added to a cytosine that is upstream to an adenine, thymine or another cytosine, and thus these modifications are referred to as CPA, CPT or CPC methylation. Non-CpG methylation has been reported in mammals in various cell types, most notably in the gene body of actively transcribed gene in stem cells and glial cells at specific stage during development. However, its functional significance in the gene regulation and disease remain poorly understood. Non-CpG methylation in male germ cells is detected that it is lost in the later stage of mitotic division. Recently, N6-adenine methylation has been reported to be crucial regulator of gene expression in embryonic stem cell and cancer cell. Mechanism of DNA methylation in gene regulation machinery involve three types of protein. First, writers. Writers are the enzyme that help to establish the DNA methylation or converting cytosine to methyl cytosine. The example of writers are DNMT1, DNMT3A and DNMT3B. Now next is the erasers. Erasers are the class of enzyme that help to remove DNA methylation, basically converting methyl cytosine back to the cytosine. Research into the DNA methylation erasers gained momentum a few years ago with the discovery of 5-hydroxymethyl cytosine, an oxidation product of 5-methyl cytosine. And it has been suggested that 5-hydroxymethyl cytosine serve as an intermediate in the DNA demethylation. It has been established that DNA methylation occurs through a series of chemical reactions which most commonly occur through 10-11 TET or TET family enzymes that further modify into the 5-methyl cytosine by deamination and oxidation reaction to a product that is recognized by the base excision repair pathway to replace the modified base. Now, next is the readers. So, readers are the class of enzymes that help to recognize DNA methylation and associated with other factors to regulate gene expression at the specific genomic locus. DNA methylation and demethylation occurs via active and passive mechanism as shown in this figure. During early development, methylation patterns are initially established by the de novo DNA methyl transferases DNMT3A and DNMT3B. When DNA replication and cell division occur, these methyl marks are maintained in daughter cell by the maintenance enzyme maintenance methyl transferases like DNMT1, which has a preference for hemi-methylated DNA. If DNMT1 is inhibited or absent due to some reason, when the cell divides, the newly synthesized strand of the DNA will not be methylated, and successive round of the cell division will result in passive demethylation. By contrast, active demethylation can occur through the enzymatic replacement of 5-methyl cytosine with cytosine. DNA methylation regulates gene expression by two means. First, it may itself reduce gene expression by impairing the binding of transcriptional activators, or with the help of several other proteins which specifically bind the methylated genomic region and set up other repressive epigenetic marks like histone methylation, histone phosphorylation, and for gene transcription to occur, the gene promoter should be readily accessible to transcription factors and other regulator units, example enhancers. DNA methylation can directly prevent transcription factor binding and lead to change in chromatin structure that restrict access of transcription factor to the gene promoter, as shown in this figure. For example, methylated CPGs attract methyl CPG binding domain protein or MBD proteins that recruit repressor complex, resulting in histone modification. Histones are the key protein component of chromatin, as we know that, and they act as spools for the wrapping of DNA which can be altered. The modification of histones by the recruitment of repressor complex lead to a more condensed chromatin structure called heterochromatin as opposed to an open and active chromatin structure we called euchromatin required for the transcription. Epigenetic changes are common in human cancer cells. Initially, most attention was paid to the DNA hypomethylation that is very common in cancer cells. This may promote tumorigenesis by transcriptional activation of proto-oncogenes which promote oncogenic cell growth, loss of imprinting or genomic instability by allowing the activation of repetitive elements like line elements, LU elements which are normally repressed, and gain of CPG methylation can also be feature of cancer cells, and as methylated cytosines are highly unstable basis, this will predispose to gene mutation as the methylated cytosine are often deaminated and converted to thymine. Replacement of a cytosine by a thymine can lead to inactivation of tumor suppressor gene, as shown in this figure. Epigenetic landscape of tumor cell is highly altered than their normal counterparts, which are established as a hypermethylation of tumor suppressor and hypomethylation of oncogene. Although a global hypomethylation signature is most commonly found in high-grade tumors. In addition, recent research has highlighted that a major consequence of epigenetic alteration in cancer cell is acquired promoter methylation, causing silencing of expression of key tumor suppressor genes. These tumor suppressor genes, example P53, P16, BRCA, etc., are required for regulation of normal growth and differentiation. Further, for targeting DNA methylation in therapy, various demethylating agents like azacitidine are used to re-express tumor suppressor and tumor specific antigen to be exploited for immunotherapy. Most epigenetic disorders which are commonly found in cancer are attributed in part to DNMT dysfunction. Defective DNMTs induce imbalances in DNA and histone modification, thus resulting in chromatin remodeling, genomic instability and gene inactivation. Recent studies have provided importance of gene-body methylation in gene regulation and its important role in cancer. There are several methods available to determine the methylation status of DNA samples. This cartoon figure summarizes the schematic representation for choosing the particular methods depends on the type of study. Different approaches provide information on DNA methylation at different levels, spanning from genome-wide methylation contents to methylation of a single residue in specific gene. All of the currently available techniques for analyzing DNA methylation are based on one of the three techniques. Example, first, bisulfite conversion. Second, digestion with methylation sensitive restriction enzymes. And third is affinity purification of methylated DNA. Several restrictions enzymes preferentially binds and cuts to recognize the sequence with preferentially methylated or unmethylated CPGs. The most common used methylation sensitive restriction enzymes are the isochimers HEPA2 and MSP1, which recognize the sequence CCGG. HEPA2 is blocked by methylation of either cytosine, whereas MSP1 is blocked only by methylation of the outer cytosine. The bending pattern of DNA fragments on agarose gel serve as the output. This approach takes advantage of the methyl binding domain or MBD containing protein domains, which bind methylated CPG sites. A tagged MBD domain expressed in E. coli is affinity purified and the MBD column is subsequently used to purify methylated DNA. This is the most common method for measuring DNA methylation. Genomic DNA is treated with sodium bisulfite. The technique based on sodium bisulfite conversion are mass array, clonal sequencing, COBRA means combined bisulfite restriction analysis, methyl specific PCR or MSP PCR, methylated and pyrosequencing are used for analysis of DNA methylation. Sodium bisulfite conversion of genomic DNA to differentiate and detect unmethylated versus methylated cytosine is the gold standard for DNA methylation analysis. Sodium bisulfite treatment completed under three steps. First, sulfonation, second, deamination, and third, desulfonation. This treatment caused deamination of unmethylated cytosine to uracil while leaving methylated cytosine intact. Bisulfite conversion involves the deamination of unmodified cytosine to uracil, leaving the modified base 5-methyl cytosine and 5-hydroxymethyl cytosine as shown in this figure. This procedure can then be followed either by PCR amplification or massively parallel sequencing method to reveal the methylation status of every cytosine in gene-specific amplification or whole genome amplification. PCR amplification of converted DNA replaced the uracil with thymine analysis of the PCR product by Sanger sequencing or pyrosequencing or mass spectrometry can be used to quantify the extent of methylation at each cytosine. Bisulfite converted DNA is subjected to PCR and sequenced directly or cloned into stable vector called bisulfite clonal sequencing. After cloning, few recombinant clones are then picked and expanded, followed by plasmid isolation and Sanger sequencing done to generate the pherogram as shown in this figure. This method is more accepted because it provides resolution regarding differentiating a heterogeneous population of bisulfite converted DNA sample, as the conversion reaction is not always 100% efficient. A number of approaches exist that enable the large-scale analysis of DNA methylation. The Human Epigenome Project has used standard sequencing approach to sequence a massive amount of bisulfite converted DNA from human tissue and primary cells, and has identified a substantial number of tissue-specific differentially methylated regions called DMRs. The beads array based analysis of DNA methylation developed by Illumina is one of the most common methods and has been utilized by several large projects including TCGA. The advanced bead chip from Illumina detect approximately 450K CpG sites covering approximately 90% of the currently curated genes. DNA methylation as a cancer biomarker. Example, methylation used as a biomarker in MGMT was shown in serve as a predictive biomarker for determining the response of glioma and glioblastoma patients treated with the alkylating agent temozolomide. Second, DNA methylation based candidate clinical biomarker genes for early detections including vimentin, septin-9 and synedican-2 for colorectal cancer, glutathione S-transferase Pi1 for prostate cancer, and cyclin-dependent kinase inhibitor 2A, CDKN2A, and short stature homobox 2, SOX2 for the lung cancer. These have already reached clinical potential and for some diagnostic test kits are also commercially available in the market. Many DNA methylation biomarkers are used for genomic imprinting disorder. The examples are Prader-Willi syndrome, Angelman syndrome, Beckwith and Beckwith-Wiedemann syndromes. So dear friends, hope you understand the basic mechanism and concept of DNA methylation. Now we summarize this module. DNA methylation is a heritable covalent transfer of methyl group to the 5th carbon position of the cytosine ring. DNA methylation is regulated by family of DNA methyl transferase enzymes like DNMT1, DNMT2, DNMT3A, DNMT3B and DNMT3L. Out of these 4 enzymes, DNMT1 is called maintenance methyl transferase. CpG islands basically is a GC rich region and around 1 kb upstream of the start site with higher CpG density. CpG islands are located as far as 2 kb from the CpG islands and CpG self located as far as 4 kb from the CpG island. DNA methylation machinery involves 3 types of protein: writers, erasers and readers. Epigenetic landscape of tumor cell is highly altered than their normal, which are established as hypermethylation of the tumor suppressor and hypermethylation of oncogene. There are several methods are available to determine and analyze a methylation pattern in the gene like methylation sensitive restriction enzyme, affinity purification, and sodium bisulfite conversion based technique like pyrosequencing, methyl specific PCR, combined bisulfite restriction analysis, and high resolution melting, etc. There are several methods are available which help to determine the methylation status in the gene. These methods are like methylation sensitive restriction enzymes, affinity purification, and sodium bisulfite based techniques like pyrosequencing, methyl specific PCR, combined bisulfite restriction analysis called COBRA, and high resolution melting etc. Thank you very much.